Produced in a process called cytolysis. And in cytolysis, the protein is inserted in the cell in this manner that creates this barrel or this tube. And then when it inserts in the membrane, it's essentially like a whole membrane. So the cell is going to rupture, and the contents of the cell are going to leak out, and that cell is going to die. So it's just giving you an example of different ways proteins can work together in a non-specific way to fight off broad range of pathogens. You also have inflammation. Okay? Inflammation and fever are relatively familiar to people, more so than cytolysis. Okay? So inflammation is a defense response against a tissue injury. Okay? So let's say you're cooking dinner and you cut your finger, and you try to put a Band-Aid on it, you put a Band-Aid on it, but it still gets infected. Okay? It gets infected, this can become inflamed. Okay? And that inflammation response serves a purpose. Okay? It might hurt, it might be warm, it might be irritating, it might be aggravating, but it limits the spread of pathogens, it destroys the pathogens that are there, removes debris and cleans up the site of infection, and initiates tissue repair and healing. Okay? So all those events are happening when you have an inflamed area. So there's 4 cardinal signs of inflammation. Okay? If you don't know these already, you may learn. I would assume that you would learn them in a nursing course as well, and use these to assess patients. Okay? So redness, swelling, heat, and pain. Okay? So the red, if the area is red, it has swelling. Heat, it's warm to the touch. When I say heat, it doesn't mean that it's hot, but it's warm. Okay? And then pain, it's painful. Okay? The redness is called erythema. Erythema comes from increased blood flow to the area of infection. Okay? Increased blood flow is called hyperemia. Swelling is called edema. That's caused by the increased leakiness of the capillaries to kind of flood the area of injury to kind of help flush things out.
Could it be like the pus that's coming out?
The swelling is more the accumulation of fluid, but the pus would contribute to the edema, swelling. The pus is usually the dead cells, bacteria, infection. And the heat comes from the increased blood flow. So the blood vessels dilate, increasing blood flow, and the area becomes red because of the blood coming closer to the surface. So that blood from the core body is going to be warm, and it's going to feel warmer to the touch. Okay? So when I say heat, I don't mean hot, I mean warm. Okay? Pain is going to be caused by chemicals that are going to be released. Inflammatory chemicals secreted by the damaged cells that are going to stimulate nerves, or can be caused by pressure on nerves. Okay? So pain is a good thing. Pain prevents you from using that area or promotes you to protect that area, okay? To promote healing. So as I mentioned, there are inflammatory chemicals that are secreted by the damaged cells. Some of those chemicals are vasoactive, meaning that they're going to promote dilation of the vessels in order to promote blood flow. If you promote blood flow, those white blood cells are going to get there faster. Okay? It's kind of like if let's say you're driving down the street and all of a sudden there's an ambulance that comes behind you. All the cars pull to the side of the road, right? To allow the emergency vehicles to get through. Similar concept where the blood vessel is going to dilate to allow more space for the white blood cells to get to the site of infection. Okay? Some of those examples of those chemicals are bradykinin, histamine, and leukotrienes. And they increase blood flow, and which increases redness and increases temperature to increase metabolism and promote healing. Okay? Another thing to note is that it increases the permeability of the blood capillaries. So the blood capillaries even become more leaky, okay, which allows blood cells, different chemicals into the tissue. Okay? clotting fences in the bacteria. So a blood clot does not form at the site of injury, but forms kind of as a fence around the site of infection. So there's a clotting fence, a fence of blood clot that kind of encircles the site of injury to trap the pathogens at the site of infection to prevent spreading of the infection. And that's what's said here, where you have the fibrinogen in tissue. It's going to clot. It's going to form a blood clot, as we described earlier, and it's going to trap the microbes near the injury. Okay? It's kind of like, let's say you have, somebody tries to break into your house, and you trap the person in the basement. Okay? You're trapping them in the basement, so you call the police. Same thing here. You're trapping the pathogens until
the big boys,
the immune system can get there to kill them. Okay? Similar situation. Heparin prevents clotting at the site of injury. So this results in the pathogens being trapped in this fluid-filled pocket at the site of injury. Okay. And then there's the process of tissue repair, where the blood, platelets, and endothelial cells in the injured area secrete different chemicals that stimulate collagen synthesis. Collagen is a protein that helps link cells together, promotes tissue repair. Hyperemia, the increased blood flow, increases the movement of materials to the site of injury and increases metabolism. Another response that's an inflammatory response is fever. Okay? We've all had a fever before. We're all monitoring ourselves for fever. Defense mechanism, it actually does more good than harm. So fever usually gets a bad rap where people think it's a bad thing. It's actually a good thing. It's your body responding to an infection. It promotes interferon activity.
Mm-hmm.
So those interferons that are those chemicals released by virus-infected cells, it actually makes them work better. Accelerates your metabolism, which is going to accelerate tissue repair. So it promotes healing, okay, if tissue has been damaged. And it inhibits pathogen reproduction. So pathogens tend not to want to replicate and reproduce in higher temperatures. Okay? So when cells are busy attacking bacteria, they secrete a chemical called the pyrogen. Okay? Pyro, think of fire, think of pyromaniac. Pyromaniac is someone that likes to start fires. Pyro means fire, heat. So pyrogen is a chemical that stimulates fever. Okay? It stimulates your body's thermostat. Your body's thermostat is in the hypothalamus. Remember your brain from AMP1, it's in your hypothalamus. A fever greater than 105 may cause delirium. Don't know who you are, what you are, where you are. Okay? Kind of confusion. Okay? If it increases to 111 to 115, even anything, 108, 107, 109.
Brain damage.
Yeah, things start to shut down, start to get organ failure, cells begin to die, things begin to shut down due to increased temperature. Okay. So if it would get that high, the patient would be, ice packs, cold cloths, wet blankets to kind of cool the body down to prevent it from getting that high. Okay. How a fever works is very similar to how, it's getting colder outside, you're going to start turning on your heat. Okay? So it works as a typical thermostat. So here it says normal body temperature, and then the infection occurs. Infection is going to occur. Those infected cells are going to secrete the pyrogen. Pyrogen is going to travel in the blood and stimulate your body's thermostat in the hypothalamus. It's going to stimulate that thermostat to turn up its set point. So the body is set at 37 degrees. Those pyrogens are going to tell the hypothalamus, we have an infection, we need a higher temperature, so you turn up the thermostat to 39 degrees Celsius. It stays at 39 during the period of infection. when the infection ends, the pathogen has been defeated, the temperature goes back down to its set point. So it's a temporary rise in temperature. But the thermostat analogy is very accurate, and that's no different than turning up the heat in your house. And then it reaches that temperature, stays at that temperature until you turn the thermostat down. Okay, so those are the routes of non-specific defenses. Okay, non-specific defenses. You also have now the specific immunity. Now the specific immunity is that third line of defense, okay, involving T cells and B cells. Characteristics that separate specific from non-specific. Specific is immunity. Immunity is specific for a particular pathogen. Okay? So we're talking about using coronavirus as a general example. So there's a big push now to get a vaccine for coronavirus. Okay? So coronavirus, if you get infected with coronavirus, the idea is your body's going to build antibodies and memory cells to coronavirus. Okay? Where if you get it, the theory is if you get exposed again, that you're going to have those defense mechanisms ready to defeat coronavirus as fast as possible so you don't get any symptoms. Okay? But there's been reports that people get infected twice. Okay? And that's because the coronavirus is thought to be a mutating virus, where the DNA mutates, where the version, if I get coronavirus, my version of coronavirus might be slightly different than somebody else's version of coronavirus. Okay? It's just like different types of shoes. Okay? Adidas, Nike, and Reebok are all shoes, just different versions of shoes, but they're all shoes, right? So there's different flavors or different versions of coronavirus that may promote a secondary infection. So that's why, because I had that question before in another class, why would I get infected twice? And that's the theory as to why that might happen. Or they're not sure how the body produces antibodies to coronavirus. Maybe the coronavirus, the antibodies don't last as long as other antibodies do. Okay, so there's a lot of research that has to be done. It's novel. That's why it's such a big deal. People really don't know much about it. This virus is meant for animals, not meant for humans. There's been a cross. Animal virus has infiltrated humans. Okay. And that's called a zoonotic disease or virus. So the immunity is specific to a particular pathogen. Memory is also a key factor. So when you're going to produce these memory cells, either B cells or T cells, and these memory cells are going to be programmed to recognize that specific pathogen and then be able to defeat it so quick you don't get symptoms. Okay? No noticeable symptoms, and that's referred to as immunity. So there's two types, as the video referred to. We have cellular immunity. Cellular immunity is mediated or regulated by T cells. Okay? Direct action on specific cells. This is cell-to-cell contact. Humoral immunity is antibody mediated, meaning that it works through antibodies. Okay? And this involves B cells. So they both are specific. You can have memory cells, B memory cells for pathogen A. You can have T memory cells for pathogen A. Just different routes of defense. Okay. Now, keeping this in mind, we have to kind of we have to understand the difference between passive and active immunity. Okay. So the video, Professor Dave talked about vaccines. Okay. Okay? And vaccines are actually a type of permanent immunity. It's flu season. So people is promoting the flu vaccine. Okay? So going through these different types of immunities to kind of keep them straight, you have natural active and artificial active. Okay? Active immunity produces memory cells. This is permanent immunity. Okay? Natural is when you're naturally exposed. Okay? So if you get naturally exposed to the flu virus, or you get a sinus infection, or you get naturally exposed to coronavirus. Okay? It produces memory cells, production of one's own antibodies or T cells as a result of a natural exposure. Okay? Artificial active will produce memory cells, but you're getting artificially exposed. So when you get the flu virus, you're being injected with inactive flu virus. Okay, they call it, the word for it is attenuated, where it's a basically dead virus or inactive virus that's going to have the antigens present though, so it's going to stimulate your immune system to produce the antibodies and memory cells, so if you do get the flu, you're not going to get any symptoms. So vaccination is kind of a proactive approach where you're going to stimulate the immune system to produce the defenses in case you do get exposed.
So you're saying that artificial active immunity is permanent production of one's own antibodies or T cells?
Right, permanent. No, it's permanent immunity, meaning that those antibodies and T cells will stick around. T cells will stick around for decades. But you may have to get a booster. So
Okay.
you get a flu vaccine every year.
Right.
To
That's
kind
like a
of
booster.
give it a boost.
Okay.
Okay? Your
Alright.
question?
Yeah, so with the flu, like with the flu every year, they always say it's a different strand every year,
Mhm.
supposedly. So why do we still flu get the vaccine though?
So if you get the vaccine,
And then there's people that still get the flu. Is it
Even if they got
a
the
different
vaccine.
strand?
different strands. So when they do the vaccines, from what I understand, is they might take the top 5 or top 10 most popular strands, or that are most prevalent, and make the vaccine from that. But there may be other strands or versions that aren't protected by the vaccine. Does that make sense?
So when you get corona, you remember how you were telling us the T and the B cells fight and then remember cells to come back type stuff. So if you get corona, can you get it again?
Yes, it's very similar to the flu.
So it don't remember those like, hey, this is a pathogen.
It morphs too quick.
See, technically,
COVID morphs too quick. It's COVID 2020 now.
in theory, if you get infected once, you shouldn't get infected again. But like the flu, there's different versions, I think.
It's
So if you
rapidly
get infected,
evolving.
yeah.
Yeah. Okay.
Yep.
So
Wasn't
you
it
could catch it
initially
if you
thought
Yeah,
that
it's the same.
it was initially thought that there was only one version,
Right.
but as time
They
went on,
made
they
this
realized
It's a mutating virus.
it's a mutation.
And they made
Yeah.
it like that.
Mutating virus, there's different versions.
Well,
If you only knew what I
coronavirus has
knew.
been around for a while, this is just a mutation of the coronavirus.
Right,
I
exactly.
know too much.
But you can
People
get
say I'm
tested,
not.
you've already had
What do you call that?
coronavirus before, you can get tested, you're supposed to have the antibodies for
Not
it.
crazy.
Right.
So it's always gonna be in your system.
Yeah. Theoretically.
Yeah.
But the antibody tests, I think, are less prevalent. I think they're harder to get right now. So if you were getting exposed and you would want to get an antibody test,
I'm getting one on the 15th. So.
You are?
Yeah.
I think they're less prevalent though. They're not
I'm
as.
just curious.
After you did the blood drive, you got tested to see if you had antibodies for it.
I'm gonna get a blood draw on the 15th to get tested for antibodies for COVID.
So I don't think it's as prevalent as.
You mean easy to access?
Yeah, it's easy to access.
Okay.
Yeah, I don't think it's as easy to access. I don't think.
As long as
And I
my
could be
insurance
wrong.
covers it and it's under,
And I could be wrong.
if they want
but
to charge
I
me
don't think it's as-
$250, I'll pay
I don't
for it.
think
I
it's easy as
got nothing
access
else going
as
for
the
me right now.
original test.
What are the
It's like
chances of the
entertainment.
flu vaccine covering the virus of the coronavirus?
It's not the same virus.
Slim.
Because
It's two different viruses.
Two different.
It is.
It's like two different animals.
it is. One is, what is flu?
Two
Flu
different.
is influenza, so what is that, like a H1N1? And I don't know, they're just different.
Yeah, they're
They have
different.
similar side effects, but they're
I'm
very
not
different.
sure.
And the COVID is morphing like crazy,
So it has different, like different versions that won't cover.
it's like a bug.
Yeah, different bugs. They're both bugs, but different types of bugs. Yep. Then you have natural and active passive. So passive is temporary immunity, right? So this is, for example, this is when the mother provides antibodies to the fetus. So the mother's immune system is giving the baby or fetus temporary immunity until the baby's immune system can be developed. Another example is, let's say you get a snake bite, or a tetanus shot, or rabies, okay? Yep.
That's fine.
Okay.
That's fine.
Okay, let's not print during class. So artificial passive injection. So this is antivenom or anti-serum. Okay, so if you get a snake bite, let's say you get bit by a rattlesnake, they'll rush you to the hospital, and they'll give you injection, which is an anti-serum or antidote, which is basically an injection of antibodies. Those antibodies are going to bind the antigens in that snake bite, which is going to give you temporary immunity, meaning it's a temporary event. You're not always going to have those antibodies in your system. So, we talked about antigens before. We talked about blood typing. We talked about A, B, and D antigens. And remember we said that all cells have antigens, even bacterial cells. Okay? So bacterial cells have antigens, and basically they're markers that can tell the immune system whether somebody belongs in the body or not. So they're identification markers. So any molecule that triggers an immune response. Okay? Immune system selects between self and non-self antigens. So self antigens are antigens that belong to the body. Non-self antigens are antigens from foreign cells. Okay? Normally only reacts to non-self antigens. If your body does react to self antigens, let's say your immune system attacks its own antigens, that results in an autoimmune response or an autoimmune disease, such as, example would be lupus. Lupus is an autoimmune disease. Or rheumatoid arthritis is an autoimmune disease where the body's immune system attacks the linings of synovial joints. Okay? Exposure to non-self antigen is what causes an allergic response. So if you're allergic to peanuts, there's proteins, antigens in peanuts that you have a hypersensitive reaction to, okay, which causes that allergic response. And there's variation in those because not everybody is allergic to peanuts. So
God.
genetics plays a role in that. Okay? And epitopes, there's a certain region of the antigen that stimulates the immune response. So there's a certain region or area of the antigen that specifically stimulates or binds the antibody. Now you have lymphocytes. Okay, so lymphocytes are the cell type involved with specific immunity. Okay? There's types of lymphocytes in the circulating blood. The vast majority are T cells. Okay? Then you have the B cells that produce the antibodies. And then you have the natural killer cells that are non-specific. They're kind of like the oddball relative. They're related, but not really. Okay? So they come from the same developmental lineage, but they don't act the same. Now, interleukins are chemical messengers that are hormone-like that allow communication between the leukocytes and the different cells. Okay? So they're messenger molecules that allow the cells to talk to one another. You have lymphokines that are produced by lymphocytes. Monokines are produced by macrophage. So So these are different chemicals that allow for communication between the different populations of cells. Okay? And there's lots of different interleukins. They're usually numbered. You have interleukin 6, interleukin 8. There's a whole family of them. They do different things. Then you have a structure called the major histocompatibility complex proteins, or MHC proteins. These are ID tags for self and non-self antigens. They're involved with the body, the immune system recognizing what's self and what's non-self. Okay? So, focusing on cellular immunity first, this is at the cell level. Okay? T cells attack and destroy foreign cells and diseased host cells. So this is a cell-to-cell interaction. It involves 4 classes of T cells. You have there's different types of T cells that have different jobs. So we talked about them in general terms. We talked about memory T cells. But you also have cytotoxic or killer T cells. These carry out the attack. They're the only T cells that directly attack bad cells. Okay, so they actually do the attacking, the killing. Okay? The helper T cells are kind of like the accomplice. Okay? They help the killer T cell do his job. Okay? Kind of like Batman and Robin.
Yeah, I
Okay?
was just gonna say that.
So helper T cells promote T and B cell activation and non-specific defense mechanisms. So they help enhance the response. Suppressor T cells keep the immune response in check. So they limit the attack, having a role in eliminating autoimmune disease. You don't want your immune system to be going overactive, because then it's going to start attacking things it shouldn't. So suppressor T cells suppress the immune response, meaning that it keeps it in check and prevents it from becoming overactive. Memory T cells, I mentioned, they provide immunity for future exposure. Okay? So I have this organized in a series of 3 steps. Okay? The first, you have to recognize the foreign cell. You have to recognize the foreign antigen. Then you're going to attack, and then there's going to be a memory produced. Okay? So you have to recognize the antigen presentation and T cell activation. So the infected cell is the antigen presenting cell is going to find an antigen, it's going to process that antigen, and it's going to present the antigen on the surface of the cell. Okay? It's kind of like how like in Lion King, where he was kind of like presented in the sky. It's the same kind of thing, where the antigen is presented on the surface of the cell. Okay? And then that antigen is processed and then recognized by the immune system. It migrates to the lymph node, displays the antigen to the T cells. Okay? The T cell checks it twice to make sure that it's actually a foreign antigen, and then it stimulates that immune response. Clonal selection occurs where there's cells divide via mitosis, and the clones to the same antigen, meaning that there's cells multiply that are going to defend against that antigen. Some carry out the attack, some become memory cells. Now, when they attack, the attack involves the helper and cytotoxic T cells. Okay? The T helper cells recognizes the antigen the MHC complex, okay, and secretes interleukins. Okay? And there's animations that I have posted that will help bring some of this into motion. They attract neutrophils and natural killer cells. They attract macrophage to come clean up debris. They stimulate T and B cell mitosis. So those helper cells enhance the immune response. The cytotoxic T cells directly attack and kill other cells. Okay? They have different ways of doing that. They have perforin and granzymes. These are chemicals that are released that kill cells as natural killer cells do. Interferons inhibit viral replication, and tumor necrosis factor aids in macrophage activation, meaning that there's different chemicals that are released that are going to have different impacts on killing that infected cell. Memory, then you have the development of those memory T cells. Okay? They're long-lived, they're more numerous than young T cells. They have fewer steps to be activated, so they're programmed to respond faster, so there's less time for symptoms to develop. Okay? T cell recall response destroys a pathogen so quick no illness occurs. Okay? So, as I mentioned, you can have this recognition, attack, and memory. You might want to help you remember, you could change it to recognize, react, and remember, the 3 R's. Okay? So you don't have to memorize all the details. Use that study guide for chapter 21 to kind of help you focus on what you need to know for the exam and help simplify the information. Now, humoral immunity, remember, works through antibodies. When you talk about humoral immunity, you're talking about B cells. Okay? The antigen binds several surface antibodies or receptors on B cells. The antigen is taken into the cell through endocytosis. It digests the antigen, okay, and displays it on the surface of the cell. Okay. T helper cell binds and secretes interleukins to activate the B cells. So the T helper cells actually activate the B cells as well. And then you get clonal selection, meaning that the B cells are going to divide via mitosis, so they're all the same. And then plasma cells, which are a special type of B cell, they are the antibody-producing cells. They produce the antibodies, and the antibody is going to be specific for that antigen. Antibodies we have talked about previously. We talked about blood typing. Okay? It's a protein. It's a large protein that's Y-shaped, okay? And they bind to antigens. Okay? They're also known as Igs or immunoglobulins. And you have this yellow region here. These yellow regions are the regions that are going to bind the antigen. Those are the most variable region. We have different types of antibodies that have different roles. So you have IgMs. IgMs are the ones that we've been talking about. Primary immune response, and they're involved with blood typing. Okay? You also have IgG. They're the majority of circulating